Well, I work with a mosquito-borne disease called malaria. About 3.4 billion people are at risk of this disease every year. There's about 216 million cases a year and about 450,000 deaths. And so new technologies are needed urgently, and the ones that we're developing in our lab are basically genetically engineered mosquitoes that are resistant to malaria. We build genes that would make the mosquito resistant to malaria parasites, and we use the CRISPR-Cas9 system to integrate those in them and then to continue to spread them. So basically what we have are synthetic anti-malarial genes coupled to this drive system. And we think the advantages of this are that it's sustainable, low cost, and resistant to the movement of mosquitoes into an area. We don't expect our technologies alone to be sufficient to eradicate malaria, but we think in combination with the development of vaccines, careful use of drugs, and other measures that we can have a role to play in that.
A little bit about DARPA, very briefly, is that we are in the business of technological surprise in the context of national security. So we want to both be able to understand and create technological surprise, but also prevent that technological surprise. And for me, the advent of CRISPR-Cas9 was a moment where I was very excited about the potential for the good uses of those technologies, but also acknowledged that there is a potential for nefarious misuse and also accidental misuse of those tools. And so really the Safe Genes program is meant, we can actually put a slide up here that shows the different aspects that the program is aimed to address. These are very powerful tools that there are good uses for them, but we want to make sure that we protect against their accidental or nefarious misuse. And so we've grouped the program into 3 technical areas where we saw there were capability gaps that existed, and we wanted to fund researchers to address those gaps and create new technical tools to better control genome editors. We want to turn on a genome editor in the cells and tissues where we want them, when we want them, and once they've turned over their target, to be removed from that system. We also wanted to generate first-in-class countermeasures and inhibitors of genome editors. So at the start of this program, there was no way to shut down an editor if there was an activity that was unintended or unwanted. And we are developing and have funded groups to look at small molecules that can inhibit genome editing activity, as well as nucleic acids, so other DNAs and RNAs that can shut down CRISPR-Cas9 activity. And then finally, we're exploring the space of genetic remediation, and this is actually correcting an edit that may have been introduced that's unwanted and restoring that system back to its baseline state. And this is certainly one of the most ambitious aspects of this program. But collectively, if we're able to make progress to control, counter, and reverse the effects of genome editors, that can give us more confidence in moving forward for applications like therapeutic uses or gene drives, which is the focus of this discussion.
I'm interested in the mosquitoes that transmit viruses like Zika virus, dengue viruses, chikungunya virus, yellow fever virus, that are also causing a lot of problems in the world. And along with some collaborators at Virginia Tech, we've discovered a gene that can basically confer maleness. Since male mosquitoes don't drink blood, they don't transmit any pathogens. And so we're trying to engineer this switch, this gene, so that we can develop populations that are biased towards being male. Then we'll hopefully have some effect on the problem.
Ecological systems are complex and unpredictable and sometimes very sensitive. For example, if you suppress a particular pest in an area, a more dangerous pest might come and fill that niche. Something that's more able to spread disease or to wreak havoc on ecosystems. So who gets to decide how much uncertainty we're willing to accept upon the first release of a gene-drive organism? Who has the right to have a voice in the debate? Who has the right to define what we consider as a risk or as a harm? There's not a really good system in the U.S. or really anywhere to have a central place where the broader socioeconomic and ecological harms are addressed. Regulations are very narrow and agencies have very specific mandates, and it's done in quite a piecemeal way. But where is the body that's going to include the public in these conversations, especially populations living in areas where gene drives are released? and to engage the public in this, and also to compare gene drives to other technological or more conventional options.